Hello, hope you are all safe and doing well. Well, students, in today's lecture we will be talking about incomplete dominance. See, in Mendelism, dominance is essentially complete. That is, phenotypically, dominant, heterozygous dominant and homozygous dominant, it appears the same. However, there are certain examples in genetics where the alleles of a particular character, they are not completely dominant or they are not completely recessive. In such cases, in F1 hybrid, what you get is a blend of the two alleles. And this phenomena is called as incomplete dominance, या जिसे हम blending inheritance भी कहते हैं. And there are examples of both plants and animals जिसमें हम incomplete dominance को study करते हैं. For example, plants में example है Mirabilis jalapa का, जिसे हम 4 o'clock plant कहते हैं. The other example is of Enterinum majus, which we also know as snapdragon या जिसे हम dog flower भी कहते हैं. Animals में there is a wonderful example of Andalusian fowl. So to explain incomplete dominance, I take the example of Mirabilis jalapa. Now there are two plants. Say if I cross a pure red flower colored plant with a pure white colored flower plant, so in the F1 we get a plant which is pink, right? Had it been a Mendelian genetics where the R allele is dominant over the W allele, then the F1 hybrid was supposed to be heterozygous tall, say if the trait is height. But here in this case of incomplete dominance, you get the F1 hybrid as pink in phenotype, right? So this is a contradiction to the Mendelian inheritance. Now when we cross this F1 plant, that is when we do the selfing, so I cross the two F1 hybrids and what we get is so what you get the genotypes as RW and WW. So this is phenotypically red, this would be pink, this again would be pink, and this is white, right? Now if this is the F2, right? Had this been a Mendelian trait, so if you see the phenotypic expression, say if I cross in the Mendelian, so what you would get is, right? So phenotypic ratio here in Mendelism would be 3 tall is to 1 dwarf, and genotypic ratio in Mendelism is 1 homozygous to 2 heterozygous tall and 1 homozygous recessive. But if we see the F2 phenotypic ratio and genotypic ratio in the incomplete dominance, so here the phenotypic ratio is 1 red is to 2 pink is to 1 white, and the genotypic ratio is again 1 R. So the genotypic ratio is 1 is to 2 is to 1. So if we see the Mendelism and if I see incomplete dominance here, what inference do I get? Number one, in incomplete dominance, if you see the phenotypic ratio in the F2 generation is different. In Mendelism it is 3 is to 1. However, in incomplete dominance it is 1 is to 2 is to 1. Although the genotypic ratio in both Mendelism and incomplete dominance is same. Second, there is no blending of the two alleles. R allele और W allele blend actually में नहीं हो रहा है. हम इसे blending inheritance जरूर कहते हैं because it seems that this is a blend of the two colors. However, if you see actually में दोनों alleles blend नहीं हो रहे हैं. क्यों? क्योंकि अगर आप F2 में देखेंगे तो आपको दोनों पेरेंट्स द प्योर रेड एंड द प्योर वाइट रीअपियर कर रहे हैं. अगर ये ब्लेंड होते सो यू वुड नॉट हैव गॉट दीज red and white flowers in the F2 generation. So there is no actual blending. Third, this also tells us about the quantitative expression of the gene. How? See, I do it here. हमारे पास 3 genotypes हैं, right? Here, what is happening is the R allele is also making its product, and this allele is also making its product. So you have double copy of the product, and hence the color is intense and it is red. In this case, this allele is not expressing and is not producing the pigment. Likewise, this allele is not expressing and producing the pigment. So the color of the flower is white. However, when we see the heterozygous state, here this allele produces the color, this allele fails to produce the color. So what you get quantitatively is half the copy of the pigment. So it seems to be a blend, and what you get is a pink color, right? So to conclude, in incomplete dominance, there is a contradiction from the Mendelian ratio in the F2 generation, where the phenotypic ratio is 1:2:1, different from the Mendelian ratio, which is 3:1. However, genotypic ratio in both the cases is 1:2:1, and there is no actual blending of the gene or the allele, since in F2 we get the parental phenotypes as well. Now if we see same thing, we can see by taking another example in animal, that is of Andalusian fowl. Here in this fowl you have 3 varieties. One is white fowl, right? The other one is the black fowl, and there is a blue fowl. So if we cross a white hen with a black cock, in F1 hybrid we get a blue fowl. Again, why do we get this blue fowl is, or why does it appear blue? It is because जो उसका कोट कलर है उसमें alternate stripes हैं, बड़ी closely set stripes हैं ब्लू और वाइट कलर की, जो कि उसको एक ब्लू कलर का appearance देते हैं. अगर हम इसको again F2 बनाएंगे तो F2 में हमें black, 2 blue और एक white कलर का फाउल मिलेगा, which again shows that black and white कलर blend नहीं हो रहे हैं. क्यों? क्योंकि F2 में आपको black कलर का फाउल भी मिल रहा है, white कलर का फाउल भी मिल रहा है, और आपको 2 blue कलर के फाउल मिल रहे हैं, right? So this is what is incomplete dominance. Thank you.